This conference will now be recorded.
Good morning, children. Thank you once again for coming to microbiology class. Today we're going to learn practicals, okay, applied exercises. Today our applied exercise is Staphylococcus. Case history. First obtained from the muscle abscess in the left upper arm of a 10 years old boy sent to microbiology lab for culture and sensitivity. Write your observation and give your report. Okay, so what is the question here? What is the nature of the sample? Sample is pus. From which site the pus is obtained? From the muscle abscess. Okay, so the sample here is pus, which is obtained from deep muscle abscess. Okay, now this sample is sent to microbiology lab. We have to culture it as well as we have to place antibiotic sensitivity test. First question, smear examination. You perform gram staining. Okay. What is the question given here? Second point. Write your observation and give your report. Right? So when you obtain a pus, you immediately inoculate into culture media. The next day, you look for growth. If there is growth, you process it. So the first step you have to do when there is growth on the culture media, perform drum stain and note what you got. Drum stain answer. The given smear stained with drum stain shows violet colored spherical shaped organisms arranged in clusters. Here and there, pink colored pus cells are seen. Okay, so this is the pus, right? The sample is the pus. So pus cells will be there, and the pus cells are pink in color. Now note, what are the bacteria that are present? Note the shape, note the color, note the arrangement. Can you see this? Yes. What is the color? Violet color. So these are violet color. So you can see it is as drum positive. What is the shape? Circle, spherical shape, round shape, right? So because it is round, it is a coccus. Next, what is the arrangement? They are in clusters, groups. So clusters. So what is your answer? First, you have to write about reactivity, means whether they are gram positive or gram negative. Next, shape, whether it is a coccus or a bacilli. Next, arrangement. Okay, so reactivity, shape, and arrangement. What is your answer? Gram positive, cocci in clusters. So smear examination is gram positive, cocci in clusters. Next one. Identify the media and note down the colony characters. When you get the sample, you have to inoculate into culture media. Okay, the culture media which we commonly employ is nutrient agar, McConaughey agar, and blood agar. So you have to see the morphological features of the bacteria on these three media or nutrient agar. Golden yellow colonies. Golden yellow. So, are they pigmented? Yes, pigment is golden yellow. Size 2 to 4 mm. Circular, convex, smooth, shiny, moist colonies. Okay, so nutrient agar, the characteristic type of colonies are golden yellow colored and they are moist. Next, blood agar. Blood agar 2 to 3 mm, circular, convex, smooth, shiny, moist, with or without beta hemolysis. So, the colonies may be hemolytic or non-hemolytic. Can you see this plate? Are they hemolytic or non-hemolytic? This is non-hemolytic. Okay, whereas hemolytic colonies may also be produced some strains of Staphylococcus. Next, McConkey agar, pink colored tiny 1 mm colonies. Look at the picture. What is the color? Pink. The McConkey agar, what type of a media is it? It is a differential media. Why are you saying it as a differential media? We say it as a differential media because it is able to differentiate between different organisms. McConkey agar is mainly used to differentiate between lactose fermenting and non-lactose fermenting bacteria. Lactose fermenters will be pink in color. Non-lactose fermenters will be pale, colorless or yellow color. If they are not pink, then it is non-lactose fermenting. So this is pink color. So it is lactose fermenting or non-lactose fermenting? It is the lactose fermenting. So once again, see the colony characteristics. Nutrient agar. What type of colonies? Golden yellow pigmented colonies. Blood agar, moist colonies with or without beta hemolysis. McConkey agar, tiny pink colored, that is lactose fermenting colonies. Biochemical reactions. There are two main biochemical reactions for Staphylococcus. One is Coagulase test and next one is mannitol fermentation test. Coagulase test, answer both slide and tube coagulase test are positive. Whereas mannitol fermentation test, it is fermented with acid only but no gas. Okay, so how do you know that the coagulase test is positive? Please look and observe the picture properly. Top tube is coagulase positive. Down one is coagulase negative. Tilt the tube. When you are tilting the tube, top tube does not overflow because it has clotted inside. Whereas down one, the liquid flows because there is no clot. So, presence of clot indicates that it is coagulase positive. No clot, coagulase negative. Antibiotic sensitivity or antibiogram. How you evaluate this? For antibiotic sensitivity testing, the media which we employ is Muller-Hinton agar. Okay? And take an HA and inoculate the bacteria. By what methods? Pour plate method. Okay? And put the desired antibiotics if you want. And look for sensitivity. Can you see the zone of inhibition? Say methicillin has a greater zone of inhibition. Next, ampicillin. Next, vancomycin. See the control. Okay, so what is this? If there is the zone of inhibition, you have to measure how many mm diameter of zone of inhibition is there, and you have to compare it with the standard values. and you have to know whether that antibiotic is sensitive or resistant. Commonly, Staphylococcus is sensitive to ampicillin, cefotaxime, erythromycin, amikacin, vancomycin, and linezolid. Next, report. Report finally. After doing everything, you have to report. So, which is the bacteria that you have isolated? Staphylococcus aureus or Coagulase positive Staphylococci, commonly known as COPS. Next one. What are the other tests to identify the pathogenic strain of the organism? What are the other tests we can use to know whether that strain is pathogenic or not? That Staphylococcus strain is pathogenic or not. First one is Coagulase test. Next, mannitol fermentation test. Next, beta hemolysis. Next, golden yellow pigmentation, gelatin liquefaction, phosphatase test, black colored colonies on potassium telluride medium, and production of thermostable DNAs. So, all of these tests are used to know if the strain is pathogenic or not. Next, you have to draw all these diagrams in your records. Okay, see the diagrams: nutrient agar, blood agar, McConkey agar, antibiotic sensitivity plate. For all of you students, please take this photo. Take a snap of this to go and do it. Okay, so draw 4 circles. To the first circle, you color it as cream color, but call this yellow color. That is nutrient agar, right? Nutrient agar, golden yellow pigment colonies. Second circle is blood agar. So blood agar, you have to color all the media red color, and colonies put it yellow color. That is hemolytic colonies. McConkey agar, full media, color it light pink. Colonies deep pink. Fourth circle, antibiotic sensitivity plate. Color it yellow and leave some circles uncolored. Those are antibiotic sensitivity. Next, 2 tubes. One is straight and one is tilted one. Straight one tube, mannitol fermentation. Okay, and inside there is a small Durham's tube, colored completely with pink color. Don't put any bubbles inside because mannitol fermentation with acid only but no gas. Next, tilted tube, that is for Coagulase test. It is positive. Liquid does not flow when the tube is tilted because it gets caught. Okay, understood all of you? Okay, so now once again come back to the following. So what have you known again? So this is Staphylococcus. So what is the question here? Pus obtained from a deep muscle abscess. So in microscopy, your answer is violet colored spherical shaped organisms. So violet colored spherical shaped organisms means what's the answer? Gram-positive cocci. Arrangement clusters. So gram-positive cocci arranged in clusters. Okay. Next, you have to draw this diagram. Gram staining, gram-positive cocci in clusters. Next one, colony characters. Nutrient agar. What type of colonies? Golden yellow moist color colonies. Next, blood agar. Large, smooth colonies with or without beta hemolysis. Next, McConaughey agar. Tiny pink color colonies. So pink color colonies means lactose fermenting colonies. Next one, biochemical reaction. So biochemical reactions. First one is Coagulase test. It is Coagulase positive. For slide and Coagulase test will be positive. Next one is mannitol fermentation test. Mannitol test is also positive, but only acid is produced, no gas formation. Next, antibiogram. Sensitive ampicillin, cefotaxime, erythromycin, amikacin, vancomycin, and linezolid. Report Coagulase positive Staphylococcus, that is Staphylococcus aureus. So what are the other tests performed to know if that Staphylococcus strain is pathogenic or not? The different tests are Coagulase test, mannitol fermentation test, beta hemolysis, golden yellow pigmentation, selective liquefaction, phosphatase test, black colonies on potassium telluride medium, and prediction of thermostable DNase. Next, please all of you draw these diagrams in your practical record. Next one. Viva voice. These are the questions which are most important for you. These questions they will ask you during Viva voice regarding Staphylococcus aureus. First question. Which points favor for the presumptive diagnosis of Staphylococcus aureus species? Presumptive diagnosis means what? Immediate diagnosis, first diagnosis. By knowing which test you can say that it is Staphylococcus species. Answer. Colony morphology. You look at the colonies and see what type of colonies are there. Pigment production. You look at the pigment. Okay, on which plate will you look for pigment? Nutrient agar. So Staphylococcus aureus, what type of pigment is there? Golden yellow pigments. There are so many pigments produced by Staphylococcus. Golden yellow pigment, white pigment, lemon yellow pigment. Among this, golden yellow is produced by Staphylococcus aureus. White, albus, lemon yellow, citrus. So, by observing the type of pigment, we can identify which type of Staphylococcus species. Next, non-motility. Next, which test is used to differentiate between Staphylococci and Streptococcus? Okay, what is this? So Staphylococci, gram staining. What is your answer? Gram positive cocci in clusters. However, many of you will get confused by saying that it is Streptococci. Then, by biochemical reactions, which test is used to differentiate between Staphylococci and Streptococci? Answer. Catalase test. So what's your answer? Catalase test. When you get a gram positive cocci, immediately perform catalase test. If catalase test is positive, then it is Staphylococcus. If the catalase test is negative, then it will be Streptococcus. Next, how will you perform catalase test? Pick a few colonies of test bacterium with a platinum loop and mix it with a drop of hydrogen peroxide on a clean glass slide. A positive catalase reaction produces gas bubbles immediately. Okay, how will you perform? You take a glass slide, okay, and add a drop of hydrogen peroxide on the glass slide. Take bacterial colonies and mix it with hydrogen peroxide. If it is positive, immediately gas bubbles will come. That is effervescence. Brisk effervescence indicates that it is catalase positive. Next question. Name some catalase positive bacteria. What are the different catalase positive bacteria? Staphylococcus species, Micrococcus species, Bacillus species, all members of Enterobacteriaceae except Shigella dysentery. So all these bacteria are catalase positive. So what are they? Staphylococcus species, Micrococcus species, Bacillus species, and all Enterobacteriaceae bacteria except Shigella dysentery. Okay, next. We have known which test is used to differentiate between Staphylococci and Streptococcus. Now, in Staphylococcus itself, there are so many species. Next point is, which test is used to differentiate between Staphylococcus aureus and other Staphylococcus species? What are the two biochemical reaction tests which we employ for Staphylococci? First one, Coagulase test. Second one, maltol fermentation test. So, among these two, one is used to differentiate. That is Coagulase test. Okay, so Coagulase test is the test which is used to differentiate between Staphylococcus aureus and other Staphylococcus species. Next, how many types of Coagulase tests are there? Answer, there are two types of Coagulase tests. One is slide coagulase, second one is tube coagulase test. Slide coagulase means on a slide, when you do this test, there will be immediate clumping. Whereas in the tube coagulase test means there will be clotting. Next, what actually is the slide coagulase test? What is the principle of slide coagulase test? Staphylococcus aureus uses an enzyme known as coagulase. It has the property to clot human or rabbit plasma. This requires plasma factor for clotting, and this plasma factor is known as coagulase reactive factor or CRF. Coagulase enzyme converts fibrinogen into fibrin. Clumping factor, that is bound coagulase, reacts with fibrinogen and causes clumping of cocci due to precipitation of fibrin on the cell surfaces. Slide coagulase tests bound coagulase. Okay, so what is the principle of coagulase test? Staphylococci produces an enzyme called as coagulase. This coagulase enzyme has the property to cause plasma, that is, human plasma or rapid plasma. However, for clotting, it requires plasma factor. Okay? The plasma factor is usually present in human or rapid plasma. So, when you add coagulase enzyme to human or rabbit plasma, the coagulase converts fibrinogen to fibrin. Clumping factor is also called as bound coagulase. It reacts with fibrinogen and causes clumping of cocci. Why there is clumping of coccus? Because due to precipitation of the fibrin. Okay. Now, bound coagulase test can be detected by using slide coagulase test. Next. So you have learned about Staphylococcus. Name the different toxins which are produced by Staphylococcus. Hemolysis, leukocidin, enterotoxin, T-acetylene, exfoliated toxin. So these are all the toxins produced by Staphylococcus. Hemolysis, alpha, beta, gamma, and delta hemolysis. T-acetylene means toxic shock syndrome toxin. So we have learned about toxins. What about the enzymes? What are the enzymes that are produced by Staphylococcus? Already you have learned one enzyme. What is it? Coagulase. Right? You have learned about coagulase. Any other enzymes are there? Yes. Phosphatase, deoxyribonuclease. All these enzymes are produced by Staphylococcus. Next, we have known about different toxins. Which toxin is responsible for food poisoning? Food poisoning by Staphylococci is caused by which toxin? Answer. It is caused by enterotoxin type A. Okay, got it? So food poisoning is caused by enterotoxin type A. Next one. Name the selective media. Which is the selective media which is employed for the growth of Staphylococcus aureus? Answer. Mannitol soft agar. Next, so we have known about Staphylococcus aureus, right? So this aureus, is it coagulase positive or negative? It is positive. So this question is name few coagulase negative Staphylococci. Answer. Staphylococcus epidermidis and Staphylococcus saprophyticus. Okay, so these are the two coagulase negative Staphylococcus. So these are the two coagulase negative Staphylococcus. Then how will you differentiate between these two? Next question. Name the test that differentiates Staphylococcus epidermidis and Staphylococcus saprophyticus. Answer. Novo biosyn test. Novobiocin is an antibiotic sensitivity test. In the case of Staphylococcal epidermidis, it is novobiocin sensitive, whereas in the case of Staphylococcus, it is novobiocin resistant. So, by observing whether sensitivity or resistance is observed to novobiocin disc, we can differentiate between gram-negative Staphylococcus species. Okay, so these are the vivae for Staphylococcus. Understood? So please all of you learn microbiology Staphylococci chapter. This will be important for practice. That's all for today's class. Thank you.